What are the key responsibilities of a clinical research monitor (CRO) during a monitoring visit, and how does their role impact the overall success of a clinical trial? Answer: a clinical research monitor (CRO) is integral to ensuring the integrity and validity of a clinical trial. Their responsibilities during a monitoring visit include review of protocol adherence. The CRA ensures that the site is following the study protocol correctly. This includes verifying informed consent processes, proper storage of investigational products, and adherence to the schedule of visits and procedures. Subject safety: ensuring the safety of trial participants is a top priority. The CRA monitors adverse events (AES) and serious adverse events (SAES) and ensures they are reported appropriately. Data integrity: the CRA reviews data for accuracy and completeness, ensuring that data entered into the Clinical Trial Management System (CTMS) is consistent with source documents, e.g., patient records. Regulatory compliance: the CRA verifies that regulatory requirements, including informed consent forms like Institutional Review Board approvals and investigator qualifications, that are in place and up to date. Communication with study team: the CRA communicates any discrepancies, corrective actions, or improvements to the site staff. They help facilitate continuous education and compliance. Impact on success: the CRO's role directly affects trial success by preventing protocol deviations, ensuring regulatory compliance, and maintaining data quality. A well-monitored site ensures that the data generated is reliable, leading to valid trial outcomes and faster regulatory approval. Explain the difference between statistical significance and clinical significance in the context of clinical research data. Why is it important to consider both? Answer: statistical significance refers to the likelihood that the results of a study are not due to random chance. It is typically measured by the p-value. A result is considered statistically significant if the p-value is less than a predefined threshold, usually 0.5. Statistical significance tells us whether the observed effect is likely to reflect a true relationship rather than occurring by chance. Clinical significance, on the other hand, refers to the practical importance of the effect size or result in real-world terms. Even if a result is statistically significant, it may not be clinically meaningful if the effect is too small to have a meaningful impact on patient care or outcomes. Clinical significance considers the magnitude of the difference and whether it is large enough to justify a change in practice or policy. While both are important, statistical significance helps determine if the study's findings are credible and reliable, but clinical significance ensures that the findings are relevant and meaningful for patient care. For example, a drug might show a statistically significant reduction in blood pressure, but if the reduction is only mHg, it may not have any real-world benefit for patients. Therefore, both measures should be considered when evaluating the results of a clinical trial. A clinical trial has completed its first interim analysis showing promising results for a new medication. The Data Monitoring Committee (DMC) has reviewed the results and suggests stopping the trial early for efficacy. What are the key ethical and regulatory considerations when deciding whether to stop a trial early based on interim results? Answer: when deciding to stop a clinical trial early based on interim results, several ethical and regulatory considerations must be taken into account. Patient safety: the primary concern is whether continuing the trial exposes participants to unnecessary risks. If interim data shows clear efficacy, continuing the trial may expose participants to a placebo or an effective treatment, which could be considered unethical. The DMC must assess the risk-to-benefit ratio for the participants. Regulatory compliance: stopping a trial early requires notification to relevant regulatory bodies such as the FDA or EMA and the Ethics Committee or Institutional Review Board according to regulatory guidelines such as those outlined in the International Conference on Harmonization (ICH). Good clinical practice (GCP) guidelines. Stopping early must be justified with robust evidence. Statistical considerations: interim analysis is conducted using pre-specified statistical methods. Stopping the trial early based on unplanned or improperly adjusted interim data could lead to incorrect conclusions. Proper methods, e.g., group sequential methods or O'Brien-Fleming boundaries, must be followed to maintain the integrity of the trial's findings. Informed consent and communication: participants must be informed about the possibility of the trial being stopped early and the implications of this on their treatment options. Clear communication with participants regarding the trial's early termination is essential for maintaining trust and transparency. The decision should balance the need to act on promising efficacy results while ensuring that participants' safety and data integrity are not compromised. Mr. Turner is a clinical research coordinator (CRC) at a site conducting a phase I trial of a new cancer treatment. During a routine monitoring visit, the CRA identifies that several patients' informed consent forms are missing signatures on the date of consent. The CRA asks Mr. Turner to address the issue immediately and recommends that he implement a corrective and preventive action (CAPA) plan. What steps should Mr. Turner take to address this issue, and how should he implement the CAPA plan in compliance with regulatory requirements? Answer: in this scenario, Mr. Turner must take the following steps to address the missing signatures on the informed consent forms. Immediate action. Corrective action. Mr. Turner should immediately review all affected ICFS and ensure that they are properly signed and dated by the patients and the study staff. If necessary, the patients should be reconsented with updated ICs, ensuring the correct dates are noted. Documentation. All actions taken should be documented in the site records, including the reasons for the missing signatures and the steps taken to rectify the issue. Identify root cause. Investigation. Mr. Turner should investigate why the signatures were missing. This might involve reviewing the consent process at the site, interviewing the study staff involved, and identifying any gaps in training or procedures. Review of procedures. The standard operating procedures (SOPs) for obtaining informed consent should be reviewed to identify any weaknesses in the process. CAPA implementation. Corrective actions. Based on the root cause, Mr. Turner should implement corrective actions, such as additional training for site staff on the informed consent process or improving monitoring procedures. Preventive actions. Preventive measures might include updating the informed consent process to ensure that all necessary signatures are obtained before the patient is enrolled in the trial. This could also include periodic audits of consent forms to ensure compliance. Regulatory and sponsor communication. Notify sponsor and regulatory authorities. The issue must be reported to the trial sponsor and relevant regulatory bodies as required by regulatory guidelines, e.g. 21 CFR Part 50 for FDA-regulated studies. A formal report should be submitted detailing the issue, corrective actions, and preventive measures. Follow-up monitoring. The CAPA plan should be followed by ongoing monitoring to ensure that the corrective and preventive actions are effective and that similar issues do not recur. Regular audits of consent forms and additional training for staff should be considered to reinforce compliance. What is the significance of power analysis in clinical trials, and how does it impact study design and sample size calculation? Answer: power analysis is a statistical technique used to determine the sample size needed to detect an effect of a given size with a certain degree of confidence. The significance of power analysis lies in its ability to determine adequate sample size. Power analysis helps ensure that the study has enough participants to detect a clinically meaningful effect. If the sample size is too small, the study may lack sufficient power to identify significant differences, leading to a type I error, failing to reject a false null hypothesis. Control for type I and type I errors. The power of a study is typically set at 80%, i.e., a 20% chance of a type I error. Researchers aim for high power to ensure that their study will accurately detect true effects. The alpha level, typically 0.5, is used to control the probability of a type I error, false positive. Effect size. Power analysis considers the expected effect size, which is the magnitude of the difference between groups. A larger effect size requires a smaller sample size to detect it, while a smaller effect size requires a larger sample size to achieve adequate power. Impact on study design. Power analysis informs the study design by influencing the number of participants to recruit, which in turn affects the resources, time, and budget allocated to the study. Explain how missing data is handled in clinical trials and discuss the statistical methods that are commonly used to address this issue. Answer: handling missing data is a crucial aspect of ensuring the integrity of a clinical trial's results. Missing data can arise for various reasons, such as patient dropout, lost follow-up, or incomplete data collection. The primary statistical methods used to handle missing data include complete case analysis. This method involves excluding participants with missing data from the analysis. While simple, this approach can lead to bias if the missing data is not random, and it may reduce the sample size, potentially affecting the power of the study. Last observation carried forward. LOCF involves replacing missing data points with the last observed value for that participant. While it preserves the sample size, it can introduce bias by assuming that the patient's condition remains unchanged. Multiple imputation. Multiple imputation involves creating several different plausible datasets based on observed data and analyzing each dataset separately. The results are then combined to produce estimates that account for the uncertainty around the missing data. This is a more robust and statistically valid method compared to LOCF. Maximum likelihood estimation (MLE). is another method used to handle missing data where the parameters of the likelihood function are estimated using all available data points. MLE can provide unbiased estimates if the data are missing at random. Weighting methods. In some cases, weighting the data by the inverse probability of missingness can help correct for bias introduced by missing data. Importance. Proper handling of missing data is critical to avoid bias and ensure the validity of the study's results. The choice of method depends on the type of missing data, e.g., missing at random, missing not at random, and the impact on the study's conclusions. What are the key differences between a NOVA and chi-square tests, and when should each be applied in clinical research? Answer: ANOVA, analysis of variance. ANOVA is used to compare the means of 3 or more groups to see if there is a statistically significant difference between them. It assumes that the data are continuous, normally distributed, and have equal variances. ANOVA is appropriate when you want to test the effect of one or more independent variables, factors, on a dependent continuous variable, e.g., comparing the mean blood pressure across 3 treatment groups. Chi-square test. The chi-square test is used to assess whether there is an association between 2 categorical variables. It is applied when the data are nominal or ordinal, and the goal is to determine if the distribution of observed frequencies differs significantly from expected frequencies, e.g., comparing the number of male and female participants across different treatment arms. When to apply. ANOVA should be used when comparing the means of multiple groups with continuous data. Chi-square should be used when assessing relationships or associations between categorical variables. How does interim analysis contribute to the monitoring of clinical trials, and what ethical guidelines govern its conduct? Answer: interim analysis involves evaluating trial data at specified points during the study before its completion. It provides valuable information about the progress and safety of the trial, guiding decisions such as whether to continue, modify, or stop the trial. Contributions to monitoring. Safety monitoring. Interim analysis helps identify any safety concerns or adverse effects early in the trial. Efficacy assessment. It can provide early evidence of treatment efficacy, potentially leading to early termination for benefit if a treatment proves highly effective. Ethical guidelines. Balancing risk and benefit. According to the Declaration of Helsinki, interim analyses should ensure that patient safety is prioritized and that any potential harm from continuing a trial outweighs the benefits. Pre-specified plans. The trial's statistical analysis plan (SAP) should define when and how interim analyses will occur, using appropriate statistical techniques to avoid inflating the type IR rate. Data Monitoring Committee (DMC). An independent DMC reviews interim data and provides recommendations, ensuring that decisions are made based on sound ethical and scientific principles. What role does site performance evaluation play in the successful conduct of a clinical trial, and how should issues with site performance be addressed? Answer: site performance evaluation ensures that clinical trials are conducted efficiently with proper adherence to the protocol and regulatory requirements. A well-performing site contributes to high-quality data, patient safety, and successful trial outcomes. Key aspects of site performance evaluation include recruitment and retention, evaluating whether the site is meeting enrollment targets and retaining participants as per the study design. Protocol adherence, ensuring that the site follows the trial protocol rigorously, minimizing protocol deviations that could jeopardize data integrity. Data accuracy, monitoring whether the site is consistently reporting accurate and timely data. Regulatory compliance, assessing whether the site adheres to all regulatory requirements, including ethics approvals, informed consent procedures, and adverse event reporting. When issues arise, they should be addressed through investigation, identifying the root cause of performance issues such as inadequate training or poor site management. Corrective action. Implementing corrective actions such as additional staff training, more frequent monitoring visits, or improved patient engagement strategies. Preventive action. Adjusting site protocols or implementing best practices to prevent future performance issues. Discuss the role of statistical software such as SPSS, SAS, or R in data analysis for clinical trials. What are the key advantages of using these tools, and what should researchers be aware of when choosing a statistical software package? Answer: statistical software like SPSS, SAS, and R play a crucial role in analyzing data collected from clinical trials. These tools are used to perform a variety of statistical analyses, including basic tests, e.g. t-tests, complex models, e.g. regression analysis, and data visualizations. Key advantages of these tools include efficiency. These software packages allow for fast processing of large datasets, which is essential for clinical trials that generate large amounts of data. Advanced statistical methods. They support complex statistical models such as multivariate analysis, survival analysis, and longitudinal data analysis, which are often required in clinical research. Reproducibility and accuracy. These tools provide consistent and accurate calculations and offer options for automating analyses, reducing human error. When choosing statistical software, researchers should consider software capabilities. The tool should be capable of handling the type of analysis required for the study, e.g., mixed effects models, longitudinal data analysis. User experience. The user interface should be appropriate for the level of statistical expertise available at the site. Explain the key differences between risk-based monitoring, RBM, and traditional monitoring in clinical trials. Discuss the advantages and limitations of each approach. Answer. Risk-based monitoring, or BM, is an approach that focuses on identifying and managing risks to the clinical trial through targeted, data-driven monitoring activities. It uses real-time data analytics, patient safety reports, and other metrics to identify potential issues and allows for more flexible, adaptive monitoring. This model aims to reduce the frequency of on-site visits and focuses resources on high-risk areas to improve trial efficiency. In contrast, traditional monitoring involves regular on-site visits by clinical research associates (CROs) to assess compliance with protocol, regulatory requirements, and data accuracy. While this method provides in-depth monitoring at the site level, it can be resource-intensive and may not be as responsive to evolving risks throughout the trial. Advantages and limitations. RBM advantages. More cost-efficient, scalable, adaptive, and focused on risk management. It increases the likelihood of identifying issues early with reduced site visits. RBM limitations. Requires advanced technology and data analytics expertise. It can be difficult to implement for smaller trials or in regions with limited infrastructure. Traditional monitoring advantages. Provides thorough oversight and direct contact with site staff. It ensures compliance with protocol and regulations. Traditional monitoring limitations. Resource-heavy, less flexible, and can miss early indicators of risks. Guidelines and regulations. The FDA encourages the use of RBM to increase efficiency as outlined in the FDA Guidance on Risk-Based Monitoring. ICHE emphasizes risk-based approaches to monitoring as a part of the Good Clinical Practice (GCP) guidelines. Describe the process for handling patient consent in a clinical trial. What are the key legal and ethical considerations when obtaining informed consent? Answer. The informed consent process ensures that patients understand the risks, benefits, and alternatives to participating in a clinical trial. This process must be conducted in a manner that respects the patient's autonomy and decision-making capacity. Key steps in obtaining informed consent. Explanation of the trial. The investigator must clearly explain the trial's purpose, procedures, risks, benefits, and the patient's right to withdraw without penalty. Comprehension. It is critical that the patient fully understands the information, which may require providing the consent form in simple, non-technical language. Voluntary decision. Consent must be given voluntarily without coercion or undue influence. Patients should feel free to ask questions and take the time they need to make an informed decision. Documentation. After obtaining verbal or written consent, the process must be documented and the patient must receive a copy of the signed consent form. Ethical and legal considerations. The Declaration of Helsinki emphasizes the necessity of obtaining informed consent for every participant in clinical trials. The Common Rule 45 CFR 46 provides the regulatory framework in the United States, requiring that consent be obtained from subjects before participation in research. Investigators must ensure that consent is ongoing, meaning patients are reminded of their rights throughout the trial. The U.S. FDA also mandates that the consent process be recorded and updated if significant changes in the trial occur. Mr. Thompson, a clinical research coordinator (ERC), is overseeing a phase II trial. During an on-site visit, he notices that the site staff has not adhered to the study protocol regarding patient randomization. The issue could potentially affect the integrity of the trial's results. What should Mr. Thompson do in this situation? Answer. Mr. Thompson should take immediate action to address the deviation from the protocol in the following steps. Document the deviation. Mr. Thompson should immediately document the specific deviation from the protocol, including the nature of the error, the staff involved, and any potential impact on the trial's results. Assess the impact. He should evaluate the potential impact of this deviation on the data and patient safety. This includes assessing whether the error has affected the integrity of the trial and if any patient safety could be compromised. Notify the investigator and sponsor. He should inform the principal investigator, PI, and the trial sponsor about the deviation and work with them to determine appropriate corrective actions. Corrective and preventive actions. CUPPA. Mr. Thompson should help develop a CAPA plan, which could involve retraining the site staff, conducting additional monitoring, and ensuring future compliance with randomization procedures. Update documentation. All actions taken, including communication with the site staff, PI, and sponsor, should be documented in the trial records. What is the role of quality management systems, QMS, in clinical research, and how do they contribute to maintaining high standards in clinical trials? Answer. Quality management systems, QMS, are essential in ensuring that clinical trials are conducted in compliance with good clinical practice, GCP, regulatory standards, and sponsor expectations. A QMS helps coordinate and streamline processes to maintain trial quality, minimize risks, and enhance data integrity. Key functions of QMS in clinical research. Process standardization. QMS ensures that processes across clinical trials are standardized to reduce variability and enhance repeatability, ensuring consistent outcomes. Risk management. By integrating risk management practices, QMS allows the identification of potential risks early in the process and helps implement mitigating actions. Documentation control. QMS ensures that all trial-related documentation, protocols, reports, data is well managed, properly versioned, and readily accessible for audits. Continuous improvement. QMS fosters continuous improvement through ongoing evaluation, audits, and corrective actions to address any deficiencies. Key regulations and standards. ISO 9001 sets the international standard for quality management and is widely applicable to clinical trial operations. Discuss the importance of team collaboration and leadership in clinical trials. How can effective leadership enhance the success of a clinical research team? Answer. Effective team collaboration and leadership are crucial to the success of clinical trials. In clinical research, trials often involve multidisciplinary teams, including investigators, coordinators, regulatory personnel, and data analysts, all of whom must work together to ensure that the trial is completed successfully and in compliance with regulations. Importance of team collaboration. Improved communication. Clear communication ensures that all team members are aware of their responsibilities and any changes to the protocol or procedures. This reduces errors and ensures consistency across the trial. Conflict resolution. Effective collaboration can help resolve conflicts between team members, ensuring that any disagreements are managed constructively without affecting the trial's progress. Resource allocation. Collaboration enables better resource planning and allocation, ensuring that necessary personnel and equipment are available when needed. Role of leadership. Strong leadership ensures that the team remains motivated, focused, and aligned with the trial's goals. Effective leaders should set clear expectations, foster a culture of accountability, provide ongoing training and support, ensure that ethical standards are maintained. Relevant guidelines. ICH, our GCP guidelines emphasize the role of leadership in maintaining trial integrity and ensuring that the team follows the protocol and regulatory standards. Effective team collaboration and leadership are also critical in the context of good clinical practice (GCP) and in ensuring adherence to regulatory guidelines. What are corrective and preventive action plans, CUPPA, and why are they critical in maintaining the quality of clinical trials? Answer. Corrective and preventive action plans, CUPPA, are systematic approaches used to address problems identified during a clinical trial and to prevent recurrence of similar issues. CAPA is part of a quality management system QMS and is critical to maintaining trial quality and regulatory compliance. Corrective action. Corrective actions are implemented to rectify a specific problem or non-compliance issue that has already occurred. These actions focus on fixing the immediate issue and ensuring that it does not escalate. Preventive action. Preventive actions aim to address the root cause of the problem, ensuring that similar issues do not occur in the future. This may involve revising procedures, retraining staff, or updating trial protocols. Why CAPA is critical? Maintains compliance. CAPA ensures that the trial stays in compliance with regulatory requirements by addressing issues that could lead to non-compliance, enhances data integrity. By fixing issues and preventing recurrence, CAPA helps maintain the integrity of the data collected during the trial, improves efficiency. Addressing and preventing recurring issues improves the overall efficiency of the trial process, reducing delays and costs. guidelines, ICHE or GCP and ISO 9001 emphasize the need for effective CAPA processes in clinical trials. The FDA also requires the implementation of CAPA in the clinical research process to ensure ongoing trial quality. What are the main factors influencing patient recruitment in clinical trials, and how can clinical research coordinators overcome challenges in this area? Answer: patient recruitment is a critical step in clinical trials, and successful recruitment ensures that the trial has sufficient participants to achieve statistically valid results. Key factors influencing patient recruitment: eligibility criteria. Strict eligibility criteria can limit the pool of potential participants, making recruitment more difficult. Awareness and education. Patients may be unaware of the trial or its benefits. Effective communication and outreach are necessary to increase awareness. Incentives. Offering incentives such as reimbursement for travel or time spent participating can help attract patients. Trust in research. Patients need to trust the research process. Building trust through clear communication and ethical practices is essential. Overcoming recruitment challenges. Simplifying eligibility criteria where possible. Enhancing outreach through multiple channels, including physicians, patient advocacy groups, and social media. Ensuring the trial is culturally sensitive and accessible to diverse populations. MS. Garcia, a CRC, is monitoring a multi-site trial and notices that one of the sites has been consistently late in reporting adverse events. How should MS. Garcia handle this situation to ensure patient safety and regulatory compliance? Answer: MS. Garcia should take the following steps to address the issue. Immediate communication. She should contact the site immediately to understand why the adverse events are being reported late and remind them of the importance of timely reporting. Document the issue. MS. Garcia should document the delay and any communication with the site staff, including the reason for the delay and any corrective actions taken. Notify the principal investigator. She should inform the principal investigator, PI, and the trial sponsor about the delay to ensure they are aware of the issue and any potential risks to patient safety. Training and remediation. MS. Garcia should arrange for training or refresher sessions for the site staff on adverse event reporting procedures to prevent future delays. Monitor for recurrence. She should closely monitor the site's future reporting to ensure that the issue is resolved. Regulatory and ethical guidelines. The ICHE or GCP guidelines emphasize the timely reporting of adverse events to ensure patient safety. What are the key components of monitoring compliance during a clinical trial, and how do these components help ensure the trial's integrity? Answer: monitoring compliance during a clinical trial is essential to ensure that the study is conducted according to the protocol, regulatory requirements, and ethical standards. Key components: protocol adherence. Ensuring that the trial is conducted as outlined in the study protocol is fundamental. Any deviations from the protocol must be documented and justified. Regulatory compliance. Ensuring that the trial follows all applicable regulations, including those from the FDA or EMA, and adheres to ethical guidelines. Data integrity. Ensuring that data is accurately and completely recorded, with appropriate documentation of any changes or corrections. Patient safety. Monitoring patient safety closely through adverse event reporting, informed consent, and continuous oversight. How these components ensure integrity. Ensuring protocol adherence reduces risks of bias or invalid results. Regulatory compliance ensures that the trial is legally and ethically conducted. Data integrity safeguards the scientific validity of the trial's results. Describe the purpose and importance of auditing clinical trial sites. What are the primary aspects that auditors should focus on during an audit? Answer: auditing clinical trial sites ensures that the trial is being conducted in compliance with regulatory requirements and the study protocol. Audits provide an objective evaluation of the site's adherence to GCP, patient safety practices, and data accuracy. Primary aspects of site audits: protocol compliance, ensuring that the site follows the approved study protocol. Regulatory compliance, verifying that the site adheres to regulatory guidelines and has all necessary approvals. Informed consent, confirming that informed consent has been obtained from all participants in the correct format. Adverse event reporting, checking that adverse events are reported promptly and documented appropriately. Importance: audits ensure that data is accurate, patient safety is prioritized, and the trial is conducted ethically and in compliance with regulatory standards. Audits also help identify any issues before they affect the study's results. What is the primary difference between a randomized controlled trial, or ET, and a cohort study, and how do these differences affect the interpretation of results? Answer: the primary difference between a randomized controlled trial (RET) and a cohort study lies in how the participants are assigned to treatment groups. In an RCT, participants are randomly assigned to either the treatment or the control group. This randomization helps control for confounding variables and biases, providing a higher level of evidence for causal relationships between interventions and outcomes. Cohort studies, on the other hand, are observational studies where groups of participants are selected based on exposure to a certain treatment or risk factor without random assignment. This lack of randomization introduces potential biases, such as confounding, making it more difficult to establish cause and effect relationships. Regulatory considerations. Good clinical practice (GCP) emphasizes the need for randomization in RCTs to ensure unbiased treatment assignment (ICA). Cohort studies require careful consideration of confounding variables and selection bias, as they do not control treatment allocation. HHS Regulations 45 CFR Part 46. A clinical research team is conducting a study on a new drug for heart disease. The inclusion criteria specify patients with a history of myocardial infarction H4570 and without severe renal impairment. What are the potential risks of having overly restrictive inclusion criteria? Answer: overly restrictive inclusion criteria can lead to several risks in clinical trials. Limited generalizability. The results may only apply to a very specific population, making it difficult to generalize findings to a broader group. Underpowered study. Restricting the criteria may reduce the sample size, increasing the likelihood of a type II failure to detect a true effect. A smaller sample may not reflect the diversity of patients who will use the drug in practice. Ethical concerns. By excluding certain populations, e.g., elderly patients with comorbidities, the trial might miss opportunities to evaluate the drug's safety and efficacy in these groups, which could lead to later risks when the drug is used in the general population. Regulatory considerations. The FDA requires that inclusion criteria ensure the safety of participants while enabling the collection of data that supports generalizable results. 21 CF R312 ICH GCP guidelines state that inclusion-exclusion criteria should ensure patient safety and align with the study's objectives. ICA. Mr. Roberts is a principal investigator, PI, conducting a study on a new cancer treatment. During the site initiation visit, CIV, the study sponsor informs him that some of the necessary regulatory documents, e.g., IRB approval, informed consent forms, are still pending. What should Mr. Roberts do in this situation, and why? Answer: in this situation, Mr. Roberts should delay any patient enrollment or study activities until the necessary regulatory approvals are obtained. The site initiation visit (CIV) is a critical step in ensuring that the site is compliant with all regulatory requirements, including Institutional Review Board (IRB) approval and the informed consent form being finalized and approved. Why this is necessary. Regulatory compliance. Enrolling patients or initiating study activities without the required approvals violates good clinical practice (GCP) guidelines and FDA regulations 21 CFR 312 and 21 CFR 50, which require that informed consent be obtained from all study participants. Ethical standards. Informed consent ensures that participants are fully aware of the study's risks, benefits, and their rights, as per the Belmont Report. Therefore, Mr. Roberts should ensure that all required documents, including IRB approval and the finalized ICF, are in place before proceeding with the study. What is the purpose of randomization in a clinical trial, and how can proper randomization affect the integrity of the study? Answer: randomization is the process of assigning participants to different treatment groups in a way that is not influenced by any human intervention or bias. Its primary purpose is to eliminate selection bias and ensure that both treatment and control groups are comparable at baseline, with each participant having an equal chance of being assigned to any group. This helps ensure that the differences observed between groups are attributable to the treatment itself, not other variables. Impact of improper randomization. Bias introduction. If randomization is not performed correctly, e.g., participants are assigned to groups based on subjective criteria, it can introduce bias, skewing the results and making them unreliable. Confounding variables. Improper randomization could result in unequal distribution of confounding factors, e.g., age, gender, or disease severity between groups, which would affect the study's internal validity. A clinical trial is investigating the use of a new anti-inflammatory drug. The primary endpoint is pain reduction, measured by a numerical pain rating scale, while the secondary endpoint is quality of life, measured by a standardized questionnaire. What should be considered when selecting appropriate endpoints for a clinical trial? Answer: when selecting endpoints for a clinical trial, the following considerations should be made. Relevance to the study objective. The endpoint must directly measure the outcome of interest. For example, pain reduction is relevant to a study investigating an anti-inflammatory drug. Measurability. Endpoints should be quantifiable and reproducible. Numerical pain rating scales and standardized questionnaires meet this criterion. Clinical meaningfulness. The chosen endpoints should have significance in terms of patient care. A clinically meaningful reduction in pain or improvement in quality of life could demonstrate the drug's therapeutic benefit. Regulatory acceptability. Endpoints must be acceptable to regulatory bodies like the FDA and EMA. Regulatory guidance often outlines acceptable primary and secondary endpoints for specific therapeutic areas. In the case of a multicenter clinical trial, what steps should be taken during the investigator site selection process to ensure that the chosen sites will provide reliable and high-quality data? Answer: during the investigator site selection process, several factors must be considered to ensure that the chosen sites will provide reliable data. Experience and expertise. Investigators should have relevant experience in conducting clinical trials, particularly in the therapeutic area being studied. Investigators should be familiar with GCP guidelines and regulatory requirements. Facilities and equipment. Sites should have adequate facilities, equipment, and resources, e.g., trained staff, patient recruitment capability to conduct the trial according to the protocol. Regulatory compliance. Sites must be able to comply with local, national, and international regulations, such as those set by the FDA or EMA, as well as institutional requirements, e.g., IRB approvals. Patient population. Sites should have access to a patient population that fits the trial's inclusion criteria and can provide the necessary sample size. Regulatory considerations. The FDA guidance on investigator selection outlines the qualifications and responsibilities that must be met. FDA 21 CFR 312.53. A clinical research team has just completed their study and is now in the process of preparing regulatory documents for submission to the FDA. What key documents should be included in this regulatory submission, and why are they important? Answer: key documents to include in an FDA submission are: clinical study report (CSO) summarizes the methodology, results, and conclusions of the study. It is vital for demonstrating the trial's integrity and the data's reliability. Investigator's brochure contains detailed information about the investigational product, including pharmacology, preclinical and clinical data, and safety information. Informed consent forms must demonstrate that the trial was conducted in accordance with informed consent requirements, ensuring ethical standards. IRB approval demonstrates that the study was reviewed and approved by an ethics committee, ensuring patient safety and adherence to ethical standards. Regulatory forms, e.g., Form 1571, required for IND submissions to the FDA to provide detailed study and investigator information. Regulatory considerations. FDA regulations 21 CFR 312 require these documents as part of the submission process to ensure that the trial has adhered to regulatory and ethical standards. GCP guidelines, ICI, stress the importance of maintaining comprehensive documentation to demonstrate trial compliance. Make a clinical trial involving a new antidepressant. The primary endpoint is the reduction in depression symptoms as measured by a scale such as the Hamilton Depression Rating Scale. What is the importance of choosing a validated, reliable measurement tool for the primary endpoint? Answer: choosing a validated, reliable measurement tool for the primary endpoint is critical for the following reasons: accuracy and consistency. A validated tool ensures that depression symptoms are measured accurately and consistently across all study sites and participants. This reduces measurement error and enhances the credibility of the trial results. Regulatory acceptance. Regulatory bodies like the FDA require that endpoints in clinical trials be measured using scientifically validated tools to ensure that the results are meaningful and reproducible. Clinical relevance. A well-established scale like the Hamilton Depression Rating Scale has been widely used and accepted in clinical trials, ensuring that the results will be recognized and understood by the medical community. Regulatory considerations. FDA guidance on endpoints in clinical trials outlines the need for using validated measurement tools for assessing efficacy in clinical trials. FDA 2016 ICHE also discusses statistical considerations, emphasizing that endpoint measures should be valid, reliable, and clinically meaningful. What factors should be considered when selecting randomization methods in a clinical trial, and what impact can poor randomization have on the trial results? Answer: factors to consider when selecting randomization methods include study design. The method should match the study design, G. Simple randomization for small trials, block randomization for larger trials to ensure balancing group sizes, sample size. Larger sample sizes may require more complex randomization methods. E.g., stratified randomization to ensure proper representation of subgroups. Bias control. The method should minimize selection bias. Computer-generated randomization is preferred over manual methods, which can introduce error. Regulatory compliance. Randomization must be conducted according to protocol and documented to comply with GCP and FDA regulations. Impact of poor randomization. Selection bias. Poor randomization can lead to unbalanced treatment groups, confounding results and reducing the study's internal validity. Inflated results. Bias can lead to an overestimation or underestimation of treatment effects, affecting the trial's reliability and potentially leading to incorrect conclusions. How should adverse events (AES) be documented and reported in a clinical trial, and what are the regulatory requirements for this process? Answer: adverse events should be documented and reported according to good clinical practice, GCP, and regulatory requirements. Documentation. AEs must be systematically documented in the clinical trial records, including details such as the onset date, severity, outcome, and relationship to the investigational product. Reporting. Serious adverse events (SAEs) must be reported to regulatory authorities like the FDA within specific timelines, e.g. 15 days for initial reports of SAEs under 21 CFR 312. Informed consent. Participants must be informed about the potential risks of AEs through the informed consent form. Regulatory considerations. FDA regulations 21 CFR 312.32 require that AES, particularly SAEs, be reported promptly to the sponsor, the IRB, and the FDA. ICHE GCP guidelines outline the procedures for AE documentation, including reporting requirements for all AEs during the trial.